Today, I'll be talking about chylomicrons. Chylos means juice and microns are small particles. Thus, these are the fat droplets. They are the largest lipoproteins composed of 8 to 9% of phospholipids, 1% cholesterol, 3% of cholesterol esters, and 85 to 86% of triacylglycerol. They are least dense because of the large proportion of triacylglycerol present. They are produced in a well-fed state, where that is intestinal epithelial cells. And why is that so? Think about its function. The conclusion can be made, which is very simple, and that is they deliver dietary lipids, which is an exogenous process. The hormone being dominant in this process is, any guesses? Insulin. After its production, it enters the blood 1 to 2 hours after the intake of a meal and accepts proteins from high-density lipoproteins becoming mature chylomicrons. All this process happens in the blood, where HDL is present. The main apoproteins being B48, C2 and E, B48 being most unique. Moving on, we have three basic stages of chylomicrons, which include nascent, mature and remnant. Now let's talk about its formation and pathway. There are 6 major structures through which this happens: the mucosal cells, blood, muscle, adipose tissue, liver and the lymph. The pathway begins with the intestinal lumen, from where it moves to the epithelial cell, then the lymph, from where it is dumped into the blood of the capillaries, from there to the muscle and adipose tissue, then finally the liver. Let's look at each structure individually. From the intestinal lumen, the digested products, fatty acid and 2-monoacylglycerol are diffused into the epithelial cells, where smooth endoplasmic reticulum forms triacylglycerol and rough endoplasmic reticulum forms ApoB48. In the Golgi apparatus, it gets modified to form the nascent or freely generated chylomicrons. Here we have the triacylglycerol with B48, nascent chylomicron. So hope everyone is following me through this process. Next, the nascent chylomicron moves to the lymph, from where it is dumped into the blood. The nascent chylomicron now requires to mature, and in order to do so, it takes C2 and E from high-density lipoprotein. High-density lipoprotein then hops off only with its AI. Now, the mature chylomicron enters the blood, which is already present in the blood, with its B48, C2 and E, which it took from the high-density lipoprotein. Also has triacylglycerol, and then it arrives to the LPL, otherwise known as lipoprotein lipase, and gets cleaved by LPL to glycerol and fatty acids. LPL, as seen, are attached on the walls of the vessel where muscle and adipose tissue are present. So what happens is that the fatty acids gets taken up by the muscle and through beta oxidation form acetyl-CoA, which by TCA cycle or electron transport chain forms ATP. Muscle thus uses fatty acid for energy. Lipoprotein lipase were activated by C2 and insulin. The fatty acid is also taken up by the adipose tissue, which activates fatty acid CoA and can join with glycerol 3-phosphate to form triacyl glycerol, which will be stored. Glycerol will be recycled by the liver. Now, when triacyl glycerol are cleaved by lipoprotein lipase, the mature chylomicrons end up becoming chylomicron remnants. It loses its B48 as well as C2. All that is left is E, which binds with ApoE receptors and is endocytosed in the liver. Once taken up by the lysosomes, fuse with it and basically break it up into different components. That was a bit too much, so let's have a quick review. The dietary sources enter epithelial cells from where nascent chylomicrons form, move through the lymph into the blood with the help of high-density lipoproteins. Mature chylomicrons are manifested. Then triglycerols are used by muscle as a source of energy and stored in adipose tissue. The chylomicron remnants goes to the liver by breaking down, and then the whole process tends to be repeated. That was all from my side. Hope that this video was helpful.